How scientists are trying to reverse death. Death has long been considered the final and absolute end of life, a point of no return. For centuries, philosophers, theologians, and scientists have debated its inevitability. But what if death is not as irreversible as we once believed? What if science could one day challenge the very nature of mortality? It sounds like something out of science fiction, but researchers around the world are actively working on methods to push the boundaries of life and death. Advances in medicine, technology, and biology are beginning to blur the line between the living and the dead, raising profound ethical and philosophical questions about the future of humanity. The science of death. What happens when we die? Before understanding how scientists are trying to reverse death, we need to define what death actually is. Traditionally, death has been seen as a single event, the moment when the heart stops beating and breathing ceases. However, modern science views death as a process rather than a sudden occurrence. In medical terms, clinical death refers to the cessation of heartbeat and breathing, but biological death, the point at which all cellular activity ceases, is more complex. After the heart stops beating, oxygen no longer reaches the body cells, and a cascade of biochemical events unfolds. The brain, which requires a constant supply of oxygen, begins to shut down within minutes. Without medical intervention, irreversible damage occurs. However, scientists have discovered that certain cells remain active for hours, even days after death. This raises an important question. If some cells can survive after the body shuts down, could death be delayed or even reversed? Cryonics, preserving the dead for a future revival. One of the most well-known efforts to defy death is cryonics, the process of freezing a body or brain in the hope of reviving it in the future. Cryonics organizations, such as Alka Life Extension Foundation and the Cryonics Institute, preserve individuals at ultra-low temperatures using liquid nitrogen. The idea is that future advancements in medical technology could one day repair cellular damage and restore life. However, cryonics remains highly controversial. Critics argue that current freezing methods cause irreversible damage to cells, particularly due to ice crystal formation. Scientists are working on advanced cryoprotectants, chemical compounds that prevent ice formation, to improve preservation techniques. While no human has ever been revived from cryonic suspension, recent studies on animals have shown promising results, keeping organs and tissues viable at low temperatures for extended periods. Reanimating the brain. The Yale brain experiment. One of the most shocking breakthroughs in the field came from Yale University in 2019. Researchers successfully revived cellular activity in pig brains hours after the animals had died. Using a system called BrainX, they circulated an oxygen-rich solution through the brains, restoring some functions, including neural activity and metabolism. Although the pigs never regained consciousness, the experiment demonstrated that brain cells could be brought back to life after death, challenging the assumption that brain damage is always permanent. This research opens the door to potential future applications, such as reviving stroke patients, preserving donor organs for longer periods, or even delaying death itself. However, ethical concerns loom large. If scientists could restore full brain function, at what point does a dead brain become a living entity again? Would a revived individual be the same person, or a different consciousness altogether? Stem cells and regeneration. Growing new organs. Another revolutionary approach involves regenerative medicine and stem cell research. Stem cells, the body's master cells capable of becoming any cell type, are being used to regrow damaged tissues and organs. Scientists have successfully grown human organs in laboratories, including miniature livers and hearts, raising the possibility of replacing dying organs with lab-grown alternatives. In some cases, regenerative therapies have already brought patients back from the brink of death. For example, cardiac stem cell treatments have helped patients recover from severe heart attacks by regenerating damaged heart tissue. If researchers can refine these techniques, they may one day be able to replace failing organs entirely, essentially reversing the effects of death at the cellular level. Hypothermic suspended animation. Pausing life to save it. A more immediate and practical approach to defying death is suspended animation. This technique, known as emergency preservation and resuscitation (EPR), involves rapidly calling the body to slow down cellular activity, effectively placing a person in a state of temporary hibernation. In 2019, surgeons at the University of Maryland successfully placed human patients in suspended animation for the first time. This experimental procedure was used on trauma patients who suffered cardiac arrest from severe injuries, such as gunshot wounds. By calling their bodies to 10-15C50-59F, doctors were able to buy precious time up to several hours to repair internal damage before restarting the heart. If this method proves successful on a wider scale, it could revolutionize emergency medicine and provide a bridge to future revival technologies, genetic engineering and longevity, stopping death. Before it starts, some scientists believe the best way to reverse death is to prevent it in the first place. Genetic engineering, particularly through CRISPR gene editing technology, is being explored as a way to slow down or even halt the aging process. Researchers have identified specific genes linked to longevity, such as the FOXO3 gene, which appears to protect against age-related diseases. Experiments on animals have already shown promising results. Scientists at Harvard have successfully reversed age-related damage in mice, making old mice biologically younger. Similar experiments have extended the lifespans of worms and FLIs by altering specific genes. If these findings can be translated to humans, genetic engineering could dramatically extend human life, reducing the risk of age-related death and potentially delaying mortality indefinitely. The ethical dilemma: should we reverse death? While the idea of reversing death is fascinating, it raises profound ethical and philosophical questions. If death can be reversed, who decides who gets revived? Would the revived person still be the same individual, or would they be fundamentally changed? How would society handle overpopulation if people could be brought back to life? There is also the question of quality of life. Even if we can restore biological functions, would a revived person suffer from cognitive impairments or loss of identity? And if cryonics or other revival methods become widely available, would only the wealthy have access to them, deepening social inequalities? The future. How close are we to defeating death? While we are still far from bringing people back to life in the way science fiction envisions, we are making astonishing progress. Technologies like cryonics, brain reanimation, genetic engineering, and suspended animation are pushing the boundaries of what we once thought was impossible. The scientific pursuit of reversing death is no longer just a dream. It is an active area of research with real, tangible breakthroughs. If humanity continues to make advances at this pace, it is possible that within our lifetime, we may witness the first successful case of revived human life. Whether through advanced medical intervention, cryonics, or genetic engineering, the future of death as we know it is uncertain. What was once considered an absolute endpoint may soon become a reversible process. And if that happens, we may need to redefine what it truly means to be alive.